So Dr. Freund was doing complement fixation?
Well, Dr. Freund was instrumental in with OP to develop the vaccine, work out the dosage, and in order to study the progress of immunization in the rabbit, the test that he used was complement fixation, and that's where I really was drilled into complement fixation. He was a very good man, Freund. That is in the laboratory and personally, but that might come later, might come up later. He taught me that, and taught me a lot of other things. He was to me a real mentor. If I perhaps a self-taught man more than anything else, but I always will remember Jules Freund as the man who really put some scientific ways of thinking in my mind. He was really exceptionally good man. I learned the test. And
then took it back to arbovirology.
I took it back to arbovirology.
Where you applied it to lopping ill?
To lopping ill and Russian spring summer encephalitis, and lo and behold, those two were very close. Now, of course, the first thing, and that's one thing that perhaps I carry on from reading about Pasteur, and Pasteur in my entire scientific life, when something happens that's exciting and new, I say, paraphrasing Pasteur, prove yourself wrong.
Yes.
And that stands one in good stead.
Did you ever try to relate St. Louis encephalitis virus?
Yes, we tried to relate St. Louis encephalitis virus with Japanese and with Russian spring summer. We never found any relationship between St. Louis or Japanese and Russian spring summer. Well, and the reason became clear later on when, after having the antigenic groups, we went into the study of the subgroups. They are a different subgroup, and you cannot find that relationship not easily by complement fixation between the tick-borne agents and the mosquito-borne. We did find some between Japanese and St. Louis. Not much, but that was because the reagents that we used at the time were not the best. The mice, the animals were immunized by means of inoculation of virus maintained in the adult mouse. And that made that the amount of virus present in the dosage was much lower than when we subsequently came into the baby, the newborn mouse, which represented a 10-fold or a 100-fold increase in the antigenic mass. And of course then the cross reactions became apparent.
And how did that happen? How did you adapt your techniques to baby mice?
Well, one of the things that we were doing at the time that I was with Peter Olitsky was to study polio, human polio, not mouse polio, although mouse polio also was part of his projects. And one thing that we wanted to do was to see whether it would be possible to develop a diagnostic test for polio. You couldn't get an antigen, an in vitro antigen, out of adult mouse brain, right, which we had used. And then it occurred to me, and in that I may have been, and I was, not I may have been, I was influenced by the work that Dr. What's his name?
Daldor?
Daldor, right, yeah. Dr. Daldor had done, in which he adapted, or isolated rather, strains of Coxsackie virus that could not be adapted or isolated in adult mice, but could very well be adapted in suckling mice, 1 or 2 or 3 days old, and that gave him at least 2 or 3 strains of Coxsackie virus.
Now, that was from
1948.
Coxsackie, New York was the first.
Yeah, Coxsackie, that's right.
First isolation.
That's right, the first isolation from the town or village of Coxsackie. Well, that made me think that perhaps one should or could attempt to maintain viruses that went very sluggishly in adult mice. What would happen if we put them in suckling mice, in 1, 2, 3-day-old mice? When that was done with the viral encephalitis, Japanese St. Louis, the result was apparent almost immediately. A shorter incubation period, an extremely high titer in the brain tissue of those animals, and in turn the titration done in a more susceptible host would host the titer to astronomical figures. Subsequently, we put the vaccine years later, the rabies virus in mice, also in baby mice. And at the time there was a colleague and friend of mine, Raul Palacios from Chile, that spent almost a year, 2 years working with me on the subject. And when he came back to Chile, he used this baby mouse passaged rabies virus to make a vaccine, and that was used for many years in Chile. It was a very successful vaccine in that the baby mouse, the suckling mouse, does not have many lipids. It's very poor in lipids. We did some work with that with Jordi Casals, and we determined that there is a period of myelinization that occurs probably between the 7th and the 10th day. Before that, the mouse brain is mostly 90% water and about 9% protein and only 1% lipid. Now, the thing that might be responsible for these accidents that follow rabies vaccination, the ascending paralysis of that, is associated with a lipoprotein which does not exist in the newborn mouse. So it was a safer vaccine. Then the concentration of virus in it was, as with any other virus, maintained in the baby mouse anywhere from 10 to 100-fold greater than in the adult mouse. So that, and then not having the lipid, it was easier to inactivate with ultraviolet light than when you had the crude suspension. So there were 3 things working with it, and they used that vaccine. Whether they still do or not, I don't know. I don't think so. And there are now better vaccines maintained in cell culture. And anyway, the process or the technique of developing vaccines is much different now. You go and get the particular protein that you want to vaccinate, and it's much better. So it's funny that the yellow fever vaccine, no matter what advances have been made, it's still the way that Max Steiler devised it, a crude suspension of virus, and nobody dares to touch it.
So by the mid-40s, you had applied the complement fixation test to arboviruses?
Yes.
And you had applied suckling mouse to
suckling mice.
Replication of arboviruses.
That's right.
And so you were preparing, really, without knowing it, to do a large amount of work in terms of growing viruses, making antigens, and comparing viruses.
That's right. Yeah, that's right. We could do that. It gave a very good test to make like a survey, like take any viruses that come and see where they fall. We did not overlook the possibilities, the fact that the neutralization test had to be employed either to corroborate the results of the complement fixation or just to determine whether there might be different antigenic moieties in the virus that might respond differently to different tests.
But in the mid-40s,
in the mid-40s,
these viruses, where they fell was usually nowhere, because they were new or unrelated to anything.
They were unrelated to anything. We began to work with viruses. I think we had 6 or 7. The St. Louis encephalitis, Japanese. Murray Valley was around, and then there was the Russian spring summer louping ill and
yellow fever.
And yellow fever. Well, the yellow fever didn't know that it belonged in that group.
Really?
In the sense that it was Albert Sabin who actually first began to talk about the relationship with yellow fever and some of the dengues. So putting all these pieces together, I had the fortune to work in an organization which became like the central depository of viruses, because the Rockefeller Foundation, in the process of studies on yellow fever, had isolated a lot of viruses, Buamba, Bunyambuera, those things, Sembliki, were just names that didn't fit anywhere, but they were just put on hold. And having begun to find relationships where relationships were not expected, it just appealed to me to see whether or not we could put some law and order in what appeared to be chaos. And the more studies were done in the tropics, or not necessarily in the tropics, the more viruses would appear that seemed to fit nowhere, and that didn't make much sense to me anyway. So it became a lumper by trying to get things in a better perspective.
And when you couldn't, then you would say subtype, or?
Yes, that's right. Well, that led to a classification, obviously, and the whole system of antigenic groups.
Well, for anyone who could figure out what a stepson-in-law is, a subtype should be easy.
That's right.
Would you like to talk about the Bunyamwera group and how that began?
Oh, the super group, the Bunyamwera group, yeah. Well, along the lines, as you know, we've talked about Group A, Group B, and other groups, Group C, and then we figured that we shouldn't run out of the alphabet. We better go into naming groups or subgroups by one of the viruses in the group. We had a discussion which really was a bit Byzantine at some time, I recall, with Max Tyler wanted to name it after the first virus isolate, and I had a hard time convinced, well, but what is the first today may be related to something that has been isolated 10 years before, and nobody had put it in this relationship. Right. So then we'll have to change the name. Oh, well, he didn't see it this way. But anyway, it became so that it was given the name of a virus in the group. The first, if so, if not the first at the time when it was named anyway. Well, after this, we, as part of our studies in trying to determine the existence of antigenic groups, we always tested, that is the fact that a virus fell in a group. It was taken subsequently as synonym with the fact that it was part of the group and of no other group. Of no other group. A virus cannot be part of two groups, antigenic groups, obviously. That was part of the thing. So that when we began to test any virus that came, this dictum, so no virus can belong into antigenic groups, was developed because not only did we test it against representative virus of a group, say Group A or Group B, as it happened to be the case, but with everything else. We never took ourselves seriously in that. Heretofore, no Group B virus, flavivirus as we call them now, can have a relationship with Eastern equine encephalitis or with Bunyamwera. But that is based not only on celebration, it's based on observation. My protocols for every single positive test that I have 100 negative ones. Mm-hmm.